Bismillahirrahmanirrahim. We start the topic-wise paper 4, chapter Photosynthesis. Now, looking at question number 1, corals grow in shallow seawater. Corals consist of colonies of small animals called polyps. So this is the small animal called a polyp, and I've made this diagram to explain this to myself. And these polyps have photosynthetic protoctists called algae that live inside their cells. Inside their cells. So these animals would have cells, and these protoctists live inside these cells, which is advantageous both to the coral polyp and to the algae. The algae that live within the cells of the polyps that can also live independently. So we have the free living ones here, which don't live inside the polyps, and we have the ones which live inside the polyps. So the algae that live within the cells of the polyps can also live independently as free living algae. So these are the free living algae, which I've just drawn here, this one and this one and this one. Now it says the rate of photosynthesis of algae that live within the cell. So these have a higher rate of photosynthesis that live within the cells of coral polyps than that of the free living. So these have a lower rate of photosynthesis. Now, I always do this to explain to myself the question, because then I can answer it more befittingly. And you can also do this homework on the side, or you can then just do it with a pencil and then rub it off, because you have to write the answer here. So suggest and explain. I've always said suggest is the most difficult part of these paper 4. So suggest and explain how living inside the cells of coral polyps increase the rate of photosynthesis. Now it makes sense. Animals, the cells will be respiring, producing carbon dioxide. So there will be abundance of carbon dioxide, plus the temperature will be the temperature of the animal. So the temperature of the carbon dioxide will not be the limiting factor anymore. And the second point would be that the coral polyps respire, so give it the carbon dioxide. And where is the carbon dioxide used? The carbon dioxide is used by the algae in the Calvin cycle. So you have to give me a benefit of the animal respiring, producing carbon dioxide, the algae photosynthesizing using the carbon dioxide, for which stage? For the Calvin cycle or the light independent reaction. Now you can see this would be the mark scheme. Increased carbon dioxide or increased temperature, because they are living inside the coral polyps and those are animals. And where are they getting this carbon dioxide? Is from the aerobic respiration of the coral polyps, and the algae will use this in the Calvin cycle. So there were 2 marks for this, and these were the 2 marking scheme points. Then we come on to the next part of the question. The relative abundance of 5 different chloroplast pigments in the algae of corals was determined. The results are shown in table 2.1. Chlorophyll A 39%, peridinin 39, chlorophyll C2 13, dinoxanthin, and beta carotene 7 and 2. So they've just given you the chloroplast pigments. Now outline the method you would use to separate and identify the pigments present in an extract of these algae. Now this is a syllabus point. So you use a chromatography or a thin layer paper, you place a spot of the extract at the base of the paper, you dry and repeat to concentrate the spot, you dip the paper in solvent so the solvent travels up, then you measure the distance traveled by the solvent and the pigment spot. And then the RF value can be calculated by the distance traveled by the pigment divided by the distance traveled by the solvent. And then the last point is you can compare the results with known RF values. So you know the pigment and you know their RF values, and then you can compare it whether it matches which of the pigments does it match. Now this is something which is a very direct question, and you have to know these steps. So you need to be figuring this out, you need to be understanding it, and you need to know how to write these steps. Then coming on to the next part of the question, table 2.2 shows the light wavelengths at which each algal chloroplast pigment shows its 2 largest peaks of light absorption. So chloroplast pigment A, peak 1 wavelength, whatever. So 430, 662, and then 456 and 485 and 450 and 396 and 442 and 471 and 454 and 480. Corals kept in tanks are often illuminated by lamps radiating mostly violet and blue light, with wavelengths in the range of 400 to 490. So the range is 400 to 490 nanometers. With reference to table 2.1 and table 2.2. So which one was table 2.1? This was table 2.1. So you refer to this and then you refer to this. When it says refer, you must see these again. Suggest why lamps radiating mostly violet and blue lights are expected to increase coral growth. Now, as it says, with reference to table 2.1 and table 2.2, suggest why lamps radiating mostly violet and blue are expected to increase coral growth. The word is increase coral growth. So pigments absorb 400 to 490 and these are 8 out of 10. You can see 1, 2, 3, 4, 5, 6, 7, 8. So 8 out of 10 peaks are between 400 to 490. There are only 2 peaks which are different. Then rate of photosynthesis of algae increases with more light absorbed. Naturally, more light absorbed, more photosynthesis. Now coral growth increases with more photosynthesis. If there's more photosynthesis, more glucose, more food, so it will increase. Then chlorophyll A and peridinin are most abundant pigments. Now that has come from this table. That chlorophyll A and peridinin are the most abundant pigment. This was another mark scheme point. Or they are the most important, because you can see that these are 39 and these are 39. Now again we have referred to table 2.1. So you must always refer to the table whenever they say it in the question. So here we will add another mark scheme point to this. So the 2 points added are number 1, chlorophyll A and peridinin most abundant pigments, and the second is 400 to 490 nanometers predominates at the depths where corals live, because if they are growing abundantly, that means the wavelength which is reaching them is causing them to photosynthesize more rapidly, so rate of photosynthesis is more. Please always read the question, understand the question, and then start writing your answers, and always look at the mark scheme points. If there are 3 points, well, you should at least give me 4 or 5, because maybe one of them you have not worded it correctly and that might be wrong. Now coming to question number 2, chloroplasts belong to a group of organelles called plastids. Although different types of plastid have different structures and functions, one type of plastid can change into another type of plastid in response to environmental or developmental signals. Example 1. Plants grown in the dark have plastids called etioplasts, which lack chlorophyll. If these plants are exposed to light, the etioplasts quickly change into chloroplasts. So now they will absorb light. Previously in the dark, they had plastids called etioplasts, which did not absorb light. Example 2. Chloroplasts in surface tissues of tomato fruits change into plastids called chromoplasts as the fruits ripen. Thylakoid membranes break down and chlorophyll synthesis stops. Chromoplasts synthesize and accumulate red lycopenes and orange beta-carotene pigments. Now it's a lot of English and a lot of understanding and a lot of very clarity of thought. A part, for each of these examples, explain the effect on the rate of photosynthesis of one type of plastid changing into another type of plastid. So explain the effect. So number 1, the rate increases as chlorophyll is needed for the light dependent reaction because they've turned into chloroplast. So that was one of the reasons why we are talking about that, how the etioplasts which were found in the dark then changed into chloroplasts. So that is, rate increases as chlorophyll needed for light dependent reaction. And example 2, rate decreases as fewer thylakoids, less chlorophyll for light dependent reaction. Here they told you thylakoid membranes break down and chlorophyll synthesis stops. Chromoplasts synthesize and accumulate red lycopene and orange beta-carotene pigments. Okay, outline the method you would use to separate and identify the pigments in an extract of tomato chloroplasts. Now this we've done in the previous question as well. I'll just quickly read through it. Chromatography, place extract on baseline of paper, dry and repeat, place paper in solvent, measure distance traveled by the solvent and the pigment, RF values distance traveled by pigment divided by distance traveled by the solvent, and then you compare the RF values against published values to identify the pigments. So you have to learn this, you have to memorize this, I'm sorry to say. There's no other way that I can explain this to you. Now let's look at the C part of the question. Cyanobacteria are prokaryotic organisms. Plastids are thought to have evolved from cyanobacteria that become incorporated into larger cells. Experiments show that free-living cyanobacteria can adapt to environmental signals in the same way as plastids. Figure 2.1 shows the absorption spectra. More the wavelength absorbed, more the rate of photosynthesis of cyanobacteria grown under two different lighting conditions. One group was grown under fluorescent light. So I have colored the fluorescent one yellow and the other group was grown under red light. So the red light I have colored it red. So I've just tried to explain this to you. The range of wavelengths absorbed by each group of cyanobacteria was then measured under identical lighting conditions. Now basically the whole question is on this graph, and if you can interpret this graph, well, you've won the game then. So if you look at the graph and look at the question they've asked you, with reference to figure 2.1 and the information given on pages 4 and 5, explain the effect of different lighting conditions on the absorption spectra of the two groups of cyanobacteria. So if you look at the graph, if you just concentrate on the graph, you see the yellow line is above it, except in this area between 580 and 660 where it has crossed it. So fluorescent lighting has more absorption. Generally it has more absorption. But in the area 580 to 660, it is the red lights which have more absorption. So look at the mark scheme points. Those pre-treated in fluorescent light have greater absorbance than those grown in red light. Well, that comes very obvious. Fluorescent light ones are more, the rate of absorption more. So just looking at the graph, just looking at something which is a very glaring point, you had to give me that. You don't have to give me any very some different sort of biology. The second point was those pre-treated in red light have greater absorbance in 580 and 660. You can see where now the yellow line is below it and the red line is above it. And the third mark scheme point was during pre-treatment, different pigments are made. So what happened is that during the pre-treatment, different pigments were made, and that is why we get these different absorbances. But because they had told you this in the beginning of the question, that when they're exposed to certain conditions, they change. And that was a question that you had to understand, and so you have to get things out of the question. So reading the question in paper 4 is a very important skill which you all must develop. Now we start with question number 3. The light dependent stage of photosynthesis when suspension of isolated chloroplasts can be investigated using the Hill reaction. You must know the difference between suspension, solution and emulsion. Suspension means something which doesn't dissolve but is suspended in it. DCPIP can be used to follow the process. DCPIP is a blue dye and turns colorless when it's reduced by accepting hydrogen and electrons. Oxidized DCPIP blue reduced is colorless. The natural hydrogen acceptor in chloroplast that is replaced by DCPIP in the Hill reaction is NADP. Reduced NADP is then used in the light independent stage of photosynthesis. So they've given you all this information, though we've taught you all this. State the role of reduced NADP in the light independent stage. Well, basically it reduces GP to TP. Now if you remember this, RUBP combines with carbon dioxide and forms a 6 carbon, then a 3 carbon, and then it is converted to 3 carbon. Now this is GP and this is TP. Now this requires 6 ATP and 6 NADPH, and then we needed to regenerate the RUBP. So we need ATP for that as well. So reduces GP to TP. Then it says predict and explain the effect on the concentration of RUBP. How will the concentration of RUBP in the chloroplast if DCPIP becomes reduced instead of NADP? So the RUBP will decrease, because what will happen is that the RUBP will combine with CO2 and will come here, and then it will stop, because it cannot go from GP to TP because there is no NADPH. We have given the hydrogens to DCPIP, so the RUBP will decrease because it's not being regenerated, because we don't have NADPH and ATP. It may be present, but NADPH is not present. So RUBP decreases because it is not regenerated. DCP-IP can be used to investigate the effect of light wavelength on the rate of the light-dependent stage of photosynthesis. One method is described here. Dip a small capillary tube into a beaker containing a suspension of isolated chloroplast. So you take a very thin capillary tube and you fill it up with the suspension of isolated chloroplast. So this is going to give you a sort of a green shade. Some of the chloroplast suspension will be drawn into tube A, forming a color standard. So you've got this color standard now for a comparison. Mix the rest of the chloroplast suspension. So the rest of the chloroplast suspension which we have in the beaker, we are going to mix it with the DCP-IP. The DCP-IP is of course a very dark blue, so you get a mixture of green and blue. And then you take another capillary tube and you mix this green and blue mixture into it. It's going to be a very dark blue mixture. It's not that light blue as I've drawn it here. So you mix the rest of the chloroplast suspension with blue DCP-IP solution. So this is chloroplast plus DCP-IP. DCP-IP plus chloroplast suspension. Dip a second capillary tube and draw up a sample to form B. So this is B and this is A, which is the green one. Store the chloroplast DCP-IP mixture for future use by wrapping the beaker. So you got this wrapped in a foil paper, covering it with a removable lid also made of foil. Now we don't want light to enter it, that's why we are covering up with foil. Now place tube A and tube B on a white tile under a bench lamp placed 15 centimeter away and immediately cover them with a purple filter as shown in figure 2.2. So measure the time taken for B to change color. So B is going to change color because A just has the chloroplast in it. To match that of tube A, this is the time taken the DCP-IP to decolorize. Repeat 4 more times using the same tube A and fresh tube B, placing the tubes under a different colored filter each time. Then we say explain the reasons for the color standard tube A. Now color standard is also C for color standard, C for comparison, C for control. To see the end point when all the DCPI has been reduced in B. But you see, how are you going to compare it whether it's greenish blue or is it green? Because green was the chloroplast and you mix the DCP-IP which was blue in it. So you got a greenish blue color. Now, how do you know that the blues become colorless? Because when it becomes the same green as the color standard A. Now, covering the beaker containing the chloroplast mixture with foil. Why did we cover this up? To stop the light entering the beaker, to stop the light-dependent reaction, to stop DCP-IP being reduced, so that all the steps start with the same color, that is the DCP-IP chloroplast mixture. But would you write all this? I mean, it's very nice of me telling you all these mark scheme points, but would you really think in these lines? This is what you have to really ask yourself. And let's look at the part C. Table 2.1 shows the results of the experiment. So color of the filter, purple, blue, green, orange, red, wavelength of light, 425, 450, 525, 625, 675, time taken to decolorize in seconds, and then rate of the light dependent stage of photosynthesis. So complete table 2.1 by calculating the rate, and this we do it by rate is 1000 over time, where t is the time in seconds. So you get an answer is 22.2, which I have then put in there. Now with reference to table 2.1, now if you just keep on looking at it, there's something which I want you to understand very clearly. With reference to table 2.1, describe and explain. So the explain is something else and describe is something else. And if you write, give me 10 explanations, I still cannot give you the full marks because you didn't do any describe. So describe, now you can see what? Describe highest at 425. This is something which you just have to look at the table. Highest is 425, this one. And the lowest is 525. This is something from the table that you have to look, nothing from your own self, nothing from what you studied in your A levels. So highest at 425 and lowest at 525. This was the describe. Describe means no biology, just give me things from the graph or the table. So this is all what you had to give me. So this would give you your 2 marks for the describe. And then the explanation, of course, is what you all know, I'm sure. Chlorophyll absorbs purple and orange light but does not absorb green. Accessory pigments are present. Light excites the electrons, and this is causes the non-cyclic photophosphorylation. And this is with the action spectrum, which tells us that different wavelengths of light have different rates of photosynthesis. So that is called the action spectrum. Now, coming to question number 4, the light-dependent stage of photosynthesis in a suspension of isolated chloroplasts can be investigated using the Hale reaction. DCPIP can be used to follow the process. DCPIP is a blue dye which turns colorless when reduced. DCPIP is an artificial hydrogen acceptor that can be used in the Hale reaction. Name the natural hydrogen acceptor found in chloroplast that is replaced by DCPIP. That's naturally NADP. Then outline the way in which hydrogen is made available to reduce the hydrogen acceptor in the light dependent stage of photosynthesis. So photolysis of water, photosystem 2 using an enzyme and hydrogen ions which have come from water and the electrons which have been excited will combine to form the hydrogen. A suspension of isolated chloroplasts for measuring the rate of the Hale reaction can be prepared by carrying out the following steps. Prepare a buffer solution with the same water potential as the stroma of the chloroplast. Liquidize or homogenize. Homogenize means you make it a homogeneous spinach leaves and ice cold buffer solution. Filter the liquid and obtain the filtrate. Centrifuge the filtrate to obtain a pellet of chloroplasts. Add the chloroplast pellet to fresh buffer solution in a beaker and mix to obtain a suspension. Explain the reasons for keeping the temperature very low, naturally to reduce activity of enzymes, because you see chloroplast would be destroyed. The chloroplast has enzymes in it, it has DNA in it, it has got an outer membrane, phospholipid bilayer, inner phospholipid bilayer. So to keep the temperatures low, we want the activity of the enzymes to be less. We don't want the lysosomes to start working. So proteases and lipases would digest the proteins in the chloroplast and the phospholipid bilayer, and this will prevent damage to the chloroplasts. So the reason why we've kept the temperatures low is just not to reduce activity of enzyme, but which enzymes? Because this was for 2 marks, so you need to give me something more about it. Part 2 says, why did you use a buffer solution? Keep the pH constant and so the enzymes work at its optimum pH. So the buffer solution was actually added to keep the pH constant. Part 3, using a solution of the same water potential as the stroma of the chloroplast. Now that was to avoid osmosis so that water doesn't enter, and so because now there's nothing really protecting it, so the chloroplast will burst and you cannot have any of the reactions taking place, and to prevent damage to the chloroplasts. Then part C, an experiment was carried out to measure the time taken for decolorization. This is just a suspension of chloroplasts and you've given the replicates. Complete table 2.1 by calculating for the 3 replicates the mean time taken. So this was the mean time, and then the mean rate, which was this one here. So we calculated that and we put it in the box as it said complete table 2.1. Now coming to the last part of this question, part 3. The time taken to decolorize DCPIP was measured at a range of light intensities. State and explain the expected relationship between light intensity and time taken to decolorize DCP. Expected relation would be as the light intensity increases, time taken for decolorization should decrease. Or you could have said as light intensity increases, rate of decolorization increases. Or you could have said as light intensity increases, decolorization is faster. So either you say light intensity, rate of decolorization increases, or you can say as light intensity increases, decolorization is faster. More the light, the more quicker the DCPIP will decolorize. Now what is the explanation for it? More light energy absorbed, faster photolysis, more electrons excited, more protons released, and a faster reduction of DCPIP. You see, for the DCPIP to be reduced, you need hydrogen. So where is the hydrogen coming from? So the electrons which have been excited by light, that is the electron, and the hydrogen is coming from the photolysis of water. So if you look at my video in which I've explained this to you in photosynthesis chapter, you will understand this relationship what I'm talking about. That completes this topic wise of the chapter on photosynthesis. Thank you very much.